of what we did, what Omar set up yesterday, and talk a little bit about working with uh, RNA-seq data in Galaxy and in ViewPath. All right, so I'm just going to start with this with a few slides. All right, so I got a lot of these slides from Catherine because I really like her genomics intro lecture. I trimmed it down a lot because we're focusing on RNA-seq. And it will be perfectly fine if anyone wants to stop and interject a question. A lot of this is, almost all of this is recap of things that have been covered at other points in the workshop, except for some of the RNA-seq specific stuff. So I'm fine. You can unmute yourself and speak, or you can type a question in the chat, and one of the other co-hosts will either answer it directly or stop me and ask the question. So I wanted to start by talking a little bit more about DNA sequencing, because RNA-seq is a DNA sequencing application, and some of the QC, some of what we're trying to make sure that it worked well is directly related to how well the DNA sequencing worked and how well the sample prep and all of that. So there are a number of different technologies that have been used for DNA sequencing over the years, but they all share some common attributes that use polymerase, which naturally copies DNA during the process of replication, but we control the polymerase, slow it down, or stop it at specific points in order to, add the bases in a controlled fashion. And we label the bases as they're being added so we can see which base is in what position. Now, when I started in molecular biology, we did DNA sequencing like this, where you ran 4 reactions for each of the different bases, and you stopped the replication or a certain percentage of the replication at each base. So you got fragments of different lengths. You labeled the last base that was added. It was both a label and a polymerase stopping base, a dideoxynucleotide. And then you ran the whole thing out on a gel. You made an X-ray image of the gel because it was labeled usually with radioactivity. And then you read them one by one and you wrote them down on your little piece of paper or right on the X-ray film. And then typically you would either read it again or hopefully you'd pass it to another student in the lab and they would read it, and you make sure that your reading was good. So, a few hundred bases a day was a pretty good rate for DNA sequencing in the 1970s and 80s. Nowadays, the vast majority of DNA sequencing is done on Illumina machines, and they use a lot of the same concepts, but one really important concept that has been added is massively parallel sequencing. So instead of taking one piece of DNA and amplifying it up enough so that you can read the bases as they're added one by one, that you get enough signal, the entire target DNA, which might be the genome or it might be cDNA made from RNA, is chopped up into pieces of an appropriate size and then all sequenced at once by spreading them out on a surface. The picture here shows kind of a conceptual representation. Actually, it looks like it's a real Illumina flow cell that has lots of tiny, tiny dots on it. False color image, obviously. And so every little dot represents a fragment of DNA that's being sequenced. Another innovation of the Illumina method is that the, the sequencing reaction is stopped after each base is added. So a given letter, an A, is added to every fragment all over the surface of this flow cell, and then it's detected, and which particular spots receive that A is recorded, and then a G is added, and the same thing happens, T and a C. So every spot should receive one of those four bases, and then the terminator is cleaved off, and another round of four bases is added, so that all of the fragments are sequenced step by step sequentially. One base is added to all the fragments, then another base. So a property of Illumina sequencing is that you get the same number of bases for every fragment that you're sequencing. So the sequence reads are all the same length. Um, yeah, blocked and labeled nucleotides are incorporated, and images taken of the entire flow cell, and it's determined which bases are being added in which position. And then that's repeated for as many cycles as you want. As Illumina sequencing has improved over the years, the precision of all of the reactions has improved, and therefore they're able to run more cycles without the signal degrading. But another feature of Illumina sequencing is that, in fact, chemistry is being conducted on millions or hundreds of millions of spots simultaneously, and there is some imperfection. It's 99.99% perfect, but some spots don't get a base added, don't have the terminator cleaved, something goes wrong. And so as the number of sequencing cycles increases, the quality of the reads goes down, the accuracy. And so we have to do some sort of quality control on the resulting sequences. You've seen this file format before. Jesse described it in the first lecture. And this is called FASTQ, which is the output from Illumina and pretty much every other sequencing technology that's operating today. So for each of those little fragments in the previous diagram, we get a read like this, which is composed of actually 4 lines of text. The first line is a header. It describes the flow cell, something about the reaction, and it tells you how long it is. Then you have the actual string of letters that were determined by the Illumina chemistry. Then this plus sign indicates that we're starting a different line. It has a repeat of the header, and then it has a line that looks kind of gibberishy. This is just ASCII text, but what it represents is one ASCII letter for each base, and that is a range from 1 to 64 that represents the quality. And by that, we mean the likelihood that the base is wrong. And so it's actually a negative exponent. So e to the minus 10 would be 1 in 10 chance that your base is wrong, and a 20 would represent a 1 in 100 chance that your base is wrong, and a 30 is a 1 in 1,000 chance that your base is wrong. The software that, well, yeah, exactly what I just said, 1 in 100, 1 in 1,000. The software that we use most frequently to evaluate the FASTQ file, obviously there's hundreds of millions of lines, you're not going to read them over and do the math on each one yourself, is called FastQC. It can read in very large Illumina FastQ files and produce a number of graphs. The one we look at the most often is just this sequence quality plot. It's in an HTML file, and it runs pretty quickly. On a typical laptop, you can probably process a 10 gigabyte FASTQ file in maybe 5 minutes. The next step in our RNA-seq workflow, after we check the quality of our sequencing, is we do an alignment. I'm just going to mention this kind of briefly. I mean, you can talk for a very long time about how alignment algorithms work, but the basic idea is that we have some bases in our genome, we have some bases in our RNA-seq read. It looks for a match, but sometimes the match is not perfect. Sometimes there is a low quality base in our read, and it's been substituted for a different letter. Sometimes there's a difference between the RNA that we're collecting from a particular organism and the reference genome for that same species. And so there'll be a mismatch. And sometimes there's sequences that are repeated in different places in the genome with changes. And so a read will map in one place with certain mismatches and in a different place with different mismatches. And the software, the algorithm has to decide what's the best place to put this read given the amount of mismatches. And our software will also, it'll have a cutoff in terms of, well, there's too many mismatches. This read just isn't going to map. And it'll also have a quality. It'll say the read goes in this position, but the possibility that it's not correct is greater than zero. There's some possibility that this read is mapped not well. After we're done with the mapping, we get an alignment of lots of reads, tens or hundreds of millions typically in a sample on the genome. Oops. And this is one way to look at them. It's probably very high resolution look, more than you would typically need when you're evaluating RNA-seq. You're going to want to skim along a lot of genes, and you don't necessarily need to have a little blue line for every single read. But it's good to know that data of this quality and this resolution is available. The mapping positions of the reads on the genome are stored in a file called a BAM file. We don't currently have a simple way for you to load BAM files from our Galaxy workflow into ViewPathDB, but they are produced. And if you want to, you can download the BAM file and then upload it into a genome browser and view the individual reads and how they line up over the genes of interest. What we do produce, obviously we also have to deal with the fact that the mRNA that we're reading in the sequencing has to align to a reference genome where introns are present. Those introns have been spliced out, and we have reads that span the intron. So the alignment algorithm for RNA-seq has to cut the reads, split them, map them on both sides of the intron, and then in the alignment software, it tends to draw a little gap like this with a thin line across, which is an intron spanning read. We introduced those when we were talking about Apollo and evaluating the evidence for introns. And so intron spanning reads are really good evidence for introns. This part of the read maps well enough here. This part of the read maps well enough here. It has to be split at exactly this position. And pretty much the only way that that could happen is when an intron is present. When you zoom out a little bit further in the BAM file, what you almost always see is that the RNA reads map mostly to where the exons have been annotated on the genome for this gene. And then often, not always, but often, you see a small amount of reads in the introns. And that is probably unspliced RNA. Either it's alternatively transcribed or we've captured some RNA before the splicing has completed, or there's some other form of read through on the transcript. If it's not stranded, you might get reads coming from transcription happening on the other strand. It's also possible to get small amount of DNA contamination in your RNA-seq reads, which may show up as reads that map inside of introns. What we do represent in ViewPath is a sort of simplified histogram where we sum up the number of reads that map over each base of the genome, and we show it in what we call a bigwig coverage track. The advantage of these tracks is that they're much, much smaller in size. And so we can store them and manipulate them and load them into JBrowse much more easily. They don't use as much RAM and as much disk storage space. And just in general, they give you most or nearly all of what you need to know about this particular RNA-seq sample and how well the reads map over these introns and exons without having to maintain every single base of every single read in a file. So the bigwig tracks is what we're going to use most of the time to evaluate our RNA-seq data. Another thing that is nice about the bigwig tracks is that this is actually the data that we're summarizing in our expression values, right? We're summing the count of bases of RNA and how many reads are covering each base of RNA in each position of exons for that gene. So essentially the sum of this histogram for that gene is what goes into our gene expression value. And then that's what we use to calculate differential expression. So what we're looking for in RNA-seq is the coverage depth, how many reads are covering each exon, differential expression between different samples. Is this gene covered a relatively different amount in a different sample? Might be a treatment, a tissue, life cycle stage, something like that. And then, as we've also discussed, we can use the RNA coverage on the genome to predict gene models. So I've circled two features of this gene model. This intron is supported extremely well by the RNA that's covering it here in the middle one. But then we see the border between these two genes, and we see that there's a fair amount of RNA coverage in a region that's not represented in the model. And so there may be untranscribed regions, UTRs, at the 3' end of the gene on the left and the 5' end of the gene on the right that are not included in our model. And if you saw this type of evidence in multiple samples, that would be a good reason to go into Apollo and produce an edited gene model that maybe includes UTRs. An example of differential expression, two different samples have very different amounts of RNA coverage on the same gene. All right, we're getting ready to jump back into Galaxy. Just recapping, Galaxy is a free web-based interface for bioinformatics tools that deal with genome scale data. So this is a way we can provide for you, our users, a relatively simple tool that doesn't involve installing software or typing command line, where you can process genome scale data such as RNA-seq and genome sequence DNA-seq. It has many, many tools, many of the most popular public domain tools. You can access public data via SRA, and you can also upload your own files that are created in your laboratory to process with our pipelines. Any command line tool or web-based service can be wrapped in the Galaxy interface. We're going to do our demo in a customized version of Galaxy that is managed for ViewPathDB, which offers preloaded genomes, private storage of your data and display, the ability to share and export results back to your ViewPathDB account into a private workspace, which we call the My Datasets section. Again, just a quick recap. The tools in Galaxy are on the left. The data, both the raw data and the products from each step of your workflow, will show up in the history on the right. And the active work panel, which is either where you're configuring a tool or where you're viewing a data file, shows up in the middle. We're going to deal with the results from a custom ViewPath workflow for RNASeq, and I'm going to be showing you a history that Omar created and shared with me. Omar showed this yesterday really well, so I'm not going to spend too much time on it today, but this is the RNAseq workflow that we're going to be looking at, where the input data are FASTQ files that come from SRA. There's two conditions that we're comparing. We run it through FastQC to produce a summary of quality. We trim off any primers or low quality bits on the RNA with Trim-O-Matic. We do the alignment to the reference genome with splicing using a tool called HiSat2. And then we count the amount of reads on each gene using a tool called HT-seq count. We also produce that bigwig file, the histogram of coverage, using BAM to bigwig. And then from those counts, we compute a differential expression between the two original conditions with DESeq2. And so I'm going to show you the result files from each of these. And Omar did all of this yesterday, and I'm going to try and jump to the QC now for our workflow. All right, so stop the share, share a different screen. Okay, so this is my Galaxy history. Typically, our workflow hides a lot of the intermediate steps and only shows a few steps that produce interesting results. So Omar's data had 3 V samples and 3 A samples. So if you click on the FASTQ of that and then on this little I button, you get the graph. I don't exactly know why this happened, but for these data files, the FASTQ did not read the quality scores properly. And I loaded another data file here. Oops. Which has a better FASTQ score. So these are not the ones that we actually have for our data, but I just wanted to show you one that was a little bit more informative. So in general, you know, this graph is divided into good, not so good, and bad. The cutoff for good is somewhere around Q30, which is 1 base in 1,000 is not correct, roughly, on average. So anything that's above Q30 means that there's less than 1 base in 1,000 wrong with the sequence at that position. And so, and then these error bars represent, the 95% confidence limit. So some bases are worse than others, but more than 95% of the bases have a very high quality score here all the way along until we get a little bit further down the read. The median, this little red line here, is still exceptionally good for all of the reads or for all of the positions on average all the way to the end. But as we get past 45 bases or so in this particular sample, there are some reads that are of very poor quality, or some bases in some reads toward the end of the read that are of poor quality. And to compensate for that, our workflow runs sequences through a trimmer called Trim-O-Matic. And Trim-O-Matic recognizes any primers that have still been stuck on the end of the sequence and might have been incorporated in the read, and trims them off. And it recognizes low quality bases and chews them off from the end of the read like a Pac-Man, so that it cuts a base. Is the quality still poor? Cuts another base, etc., until it gets to the point where the quality of that particular read is good. So it works on each individual read to trim it down to the point where the quality is good. Sometimes I rerun data through FastQC after it's been trimmed with Trim-O-Matic to double check that the reads are in much better shape now. Our workflow does not do that. We don't repeat the FastQC, but if you open up all the hidden stages of the read, you could find down here the output from Trim-O-Matic. So there's the FastQCs. There's a lot because there's 6. So this is the output from Trim-O-Matic. And it still looks like a FastQ file. And you could take this as input and run a second FastQC on it. It still has this weird property of question marks where the quality scores are supposed to be. So this particular data file probably won't benefit from another round through FastQC. So after we run Trim-O-Matic, we take the data into the aligner, HiSat2, and the output from HiSat2 is an alignment file. These are not particularly easy to read as a text file, but if you really wanted to know what it looks like, this is kind of what it looks like. There's one line for each read. It contains the read itself. It contains the position on the genome and the chromosome where that read matches. Tells you how many bases actually match the read, 76 for this one. And then there's a string over here that represents any information about mismatches and some other things about the overall quality of the alignment at that position, if there are other positions in the genome that had similar but maybe not quite as good alignment. So that's called a cigar string. Again, this is not really meant to be a human readable. It's meant to be input for the next step. The other output from the aligner is, well, this is actually, we run it through BAM to Bigwig, right? This is a BAM formatted file, or SAM, because we can read it. And then we make a Bigwig file, which is that histogram. I'm going to show in a minute how to load this back into ViewPathDB so you can view it on the proper genome. But I found this cute little visualization tool built into Galaxy, and I thought I'd show it just because it is very quick. You could name it. It correctly identifies what genome, probably because Omar annotated his files when he uploaded them to say what genome they were. And it loads the histogram for this particular chromosome. And so if you pick a chromosome that has good genes on it, you'll get to see the histograms. Unfortunately, it doesn't load the GFF file, so you can't look at the RNA reads with respect to where the gene names and exons are annotated. But just as a very quick QC, you can say, yep, my RNA-seq file aligned to the genome, and I get stacks of reads that look a lot like genes. So that level of QC was successful, and I'm happy with it. Oops. Oh, back to analyze data. All right, so that is the BAM to bigwig file. And then next step is we're gonna count the genes, count the reads on each gene. And so at this point, the output is a simple count. Oops. And a lot of genes get 0 counts, but hopefully some genes do get good number of counts in this data file. And obviously this is your raw data, right? It's not a simple histogram, it's a simple Excel table. So you could download this HT-seq count on the data as a tab delimited text file right here. You can just download it to your desktop, open it up in Excel, and sort by gene count. Or you could download the counts for every sample, sort them by sample name, and then compute for yourself the full change across the average of samples for one treatment versus the average of samples for another treatment. Or you could feed these counts into a statistics tool in R. So very useful output file that is produced by the workflow. And so there's an HT-seq count for each of our samples. Yes. And there's a bigwig file. All right. And then the last thing I want to show is the differential expression. And this tool called DESeq2 takes the counts of each sample for each gene and does a fold change between the two samples. So we have already pre-labeled the V samples and the A samples, and so it knows which one to compare, computes a log fold change for each gene between those samples, and a p-value for that comparison, and then a Bonferroni adjustment to say, okay, the adjusted p-value, given that we're testing p-values for many genes, is 1.5 times e to the 88. So, oh, and then the genes are ranked by p-value. So by default, the most significant genes will show up in the top of this tab delimited text file. So this is another file you might want to download to your desktop and save for, browsing in Excel or whatever. The other thing that DESeq2 produces is a really nice PDF file that has a good summary of the statistical information. And when I do an RNA-seq analysis, this is the first place I go to say, how well did my experiment work? We had 3 replicates of 2 conditions, and this is a PCA graph, and it's showing, when you summarize all of the expression data for all of the genes and cluster the samples by that expression data, do the samples separate by treatment groups? And in this case, they separate extremely well. Um, the the axis here, the horizontal axis, contains 65% of all the variance in the data, and these samples are separated extremely well on this x-axis. The second axis, which contains 15% of the variance, separates the samples within each group vertically to some extent, and more in the A samples than the V samples. So there's a bit more variability in the A samples, and the V samples are more similar to each other. So that's a very good thing to look at as a first step in understanding the differential expression analysis of your RNA-seq experiment. This is saying the same thing in a different way. It's saying that obviously the samples map well to themselves, but also the V samples are more similar to each other, and the A samples are more similar to each other. This graph of dispersion is basically, well, I'll just say that these things are graphed quite tightly. There's not a huge amount of variance showing up here. And so that works well. I like this graph a lot, the M versus A graph. The horizontal axis is just number of counts. So these are the genes, the dots, and the ones at the right are genes that are expressed more highly. And the ones at the left are genes that are expressed at a low level. The vertical axis is the fold change. So what you hope to see, like in this graph, is that we have differential expression that's not particularly correlated with expression level. So some high expressed genes show differential expression, and some low expressed genes show differential expression. And the ones that are extremely low, having, one or, very, very few counts per gene, are not differentially expressed because there's not enough variance there to do the calculation and show them being differentially expressed. So that's all good. The normalization and the statistical methods have suppressed variance in the very low count genes, and we get a good amount of differentially expressed genes from moderate to high expression levels. So this stuff looks all pretty good. And yeah,
Stuart, I just want to let you know we have about 10 minutes left, and there
right.
was
So now I'm going to jump over to the last bit. I mean, this is all our output. And now we want to jump back to uploading this data back to ViewPath. So we have some tools over here, ViewPath export tools, where this is RNA-seq to ViewPath. Once you've completed an RNA-seq expression, you have your BAM to bigwigs and your HTC counts. You can put in any notes that you want to put in here. You definitely want to give your data set a name, and then you just hit execute, and these data files should jump back to your ViewPath account. And oops, I lost my window for that. Well, I'm going to try and do it again. All right, I'm going to have to stop the share and share a new screen. You need to know what section of ViewPath your data belongs to. That is set up here in the export as well. No, it's just recognized by ViewPath based on the genome that you've chosen. So this one is Pyroplasma DB. And then if you go to my workspace and my data sets, the data set that I exported shows up here as my data set. You can click on it. And one of the nice things you can do is you can view it in the genome browser. And this will automatically find the correct genome, Babesia bigemina, strand BOND, and it will show you this particular file. And then you can go over here to select tracks, choose my data from Galaxy, and load up all the tracks. And I discovered this the other night, is that the A samples, whoops, they didn't line up perfectly. The A samples have pretty good coverage, at least for some of the genes, many genes, and an appropriate number of genes. But the coverage in the V samples is extraordinarily low. I can see it if we change to log scale, but I'm worried about this in terms of, oops, in terms of calculating the fold changes when all of the samples in one treatment have, about 1,000 reads covering this gene, and samples in the other treatment have 5 or 9 reads covering this gene.
One thing, Stuart, just to interject quickly, I can tell from this, I must have mislabeled samples as well. It's always good to look at your data and say, does it make sense? So this is an experiment. I don't know the details of the experiment, but from the description in the sequence read archive, they used a virulence strain of Babesia bigemini and then an attenuated vaccine strain. That's the A strains. And then they did RNA-seq on the infected infected cows, basically. And so you would expect that if the attenuated strain, the vaccine strain, is working, it's not going to grow well, and so you're going to have very little, a small amount of parasites, or a small amount of RNA in your samples in the end. And so it would make sense that from your attenuated samples, you get a small amount of RNA for your, is much more reduced. So that's why I'm concluding that I mislabeled them here.
Okay. So I'm a little worried about this because the DE-seq2 algorithm, while it does normalize for reads per, for coverage, it can't compensate entirely for very different coverage among all the samples for one treatment versus another. That's we would call it a confounding factor, and not necessarily, something that you would want. All right. One other thing I can show you, I've got like a couple minutes left, is
we also have some questions to discuss.
Cool.
So yeah, when you're done, we'll do that.
I'll do 1 minute on this and then we'll jump to the questions. You saw this yesterday, so I'm not going to do the searches right now, but your data is available for fold change search, right? So I can do A versus V and say, show me everything fold change 4 that is upregulated in A versus V, get answer, and I will get a gene list of the upregulated genes. It so happens that I have 627 genes that are considered upregulated. And this is really nice. you get this little graph that confirms that, yeah, the V samples have a lot more expression than the A samples for this gene using TPM, transcripts per million. So that's where it's been normalized. And you can click through back to that gene and get as much information as we know about that gene, whether it has gene ontology associated with it, links maybe to UniProt, other databases, if there's any curation and annotation, and if we're lucky, we might have an alpha fold, which is always very pretty. All right. We can jump to questions now. I don't know if I can see them. I'll have to stop sharing.
Yeah, that's fine. There was a little discussion going on about unique versus non-unique mapped reads and how we treat that in
Sure.
ViewPath.
So the unique mapped reads have high quality on the location where the BAM file shows they are. The non-unique reads mean that they map to more than one location on the genome. Mostly those are repetitive sequences. And when it says non-unique, basically the aligner can't really decide with good level of certainty where that gene should map. They don't need to be completely ignored, but they're not used in the HT-seq count and the DE-seq measurements for gene expression. So usually when you visualize, you most of the time want to only visualize the unique mapped bigwig files.
And that actually, to me, so I posed the question of duplicate reads, and now there's like a subtlety of unique versus non-unique within the context of duplicate reads. But I was seeing that duplicate reads are a thing that get dealt with likely prior to HT-seq count.
Yeah, duplicate reads are an artifact of the sequencing machine. I showed you those little colored dots, right? Now, sometimes the image analysis software will split a dot into two. And so you'll get two reads from the same original fragment of DNA. And we don't do it in our workflow, but you can filter out duplicate reads if you want to. It doesn't tend to make a lot of difference in RNA-seq. You tend to have pretty good coverage, thousands of reads covering each position on a well-expressed gene. And so the fact that some small number of them are duplicated, and so they show up exactly on top of each other in the alignment, doesn't make a lot of difference. In certain, yeah.
Sorry, can I just interject there? So when you do the quality control, the FastQC actually makes the assumption that you're sequencing the genome, not the RNA. And so it's making the assumption that it's very unlikely you're going to sequence the same read more than once. But if you're doing RNA-seq and you have one or a small number of genes that are very highly expressed, the chance that you sample exactly the same region more than once increases. And so it's common. It is actually a thing that happens in RNA-seq purely because you do have some genes which are sampled much more often than other genes. And so for anyone working on Trypanosoma brucei, a really good example is the active VSG, which is usually expressed at least tenfold more than any other gene in the genome. And so it's very common in that case to see duplicate reads from the active VSG just because of the biology. So it's in the case of RNA-seq, I quite often see it as a function of the biology. But if it's a case if I'm doing whole genome sequencing, then it does tend to be artifacts in the same way as Stuart is talking about.
Very true. Yeah. Also, obviously, transcripts all begin and end at the same place. So you're going to have a bunch of reads that begin at the beginning and end at the end, or begin at the end and end at the beginning. So yeah, that's a FastQC thing. It flags duplicate reads, but like Catherine just said, FastQC was designed for DNA sequencing, and you need to pretty much ignore that for RNA-seq.
Great. These are very important things to understand and especially put into context. I would just say that when you see those bigwigs and those coverage maps, I have this curiosity if like reads were like 150 base pairs, why they're, why all the histograms aren't all the way to the max level for, and I guess it's because some reads just don't, duplicate reads might throw off the histograms, but now you're saying really not too much. I don't know.
I don't know. What are you asking about the histograms? That they're not
Yeah.
equal?
Yeah, so if 150 base pair long stacked up on top of each other for each position of that base, why isn't the count equal all the way across? But you should only be having unique reads, and they should not be overlapping on top of each other. So that's what's showing the histograms the way they are, or the peaks in certain, like the midsection of a gene versus the outer regions, the coverage, read coverage seems to be shorter all the time. But some genes, it's very high across multiple bases.
And another factor here is the local GC content, because obviously sequencing does an amplification of PCR. And as anyone who's done molecular biology is aware, GC-rich sequences tend to amplify better than AT-rich sequences. So very local GC content does tend to affect the coverage as well. So it's often not like not even within a gene, just because you have short regions within that gene that amplify better.
Yeah, another aspect of this is the workflow, the lab workflow that makes your cDNA and feeds it into the Illumina workflow. So you might not, at the very ends of the gene, you're going to get really short fragments that don't get captured as efficiently by the Illumina sample prep. So you'll always see a sloping off near the ends of your transcript. And coverage will be deeper near the middle.
Okay, yeah, this is so great. Thank you so much. Really sheds a lot of light on a very subtle thing, I think, that
yeah, so
everybody
sometimes
else.
you can QC too well, right? You're like zooming in on each individual aligned read, when the TPM numbers really do capture a lot of what you need to know about the coverage of that gene. And the biases should be consistent from one sample to another, the GC content of that gene or whatever. So when you look at the ratio, the fold change or the p-value, that takes everything into account. And it's saying, after all the mapping and artifacts and whatnot, is the coverage of this gene significantly different in this sample versus this other sample?
Right. So read count and read coverage, those sound like very similar kind of things related to each other, but coverage is going to be across the extent of a region of
Correct. And count,
the genome.
yeah. Well, yeah, count is usually like base by base, and coverage is, it's normalized across the whole gene.
Wow. Okay, cool.
Okay, great. I'd suggest, Jean-Paul, if you have more questions, we could make a breakout room for you and Stuart, or we can continue this discussion at 1 o'clock when we
Perfect.
finish,
Yeah,
'cause these
sorry
are
to
great.
yeah,
I'd love
sorry
for
to
you
take
to
It's
get
great. We
everything.
love questions.
Yeah. But right now I think it's probably a good idea to move on